Hello students, today we will see how isolation of bacteria present in the rhizosphere can be done. So rhizosphere is the area around the root closely attached. In 1904, a famous scientist, Hiltner, for the first time coined the term rhizosphere to denote the area of intense microbiological activity that extends several millimeters from the root system of the growing plant. Microorganisms growing under the influence of root are often qualitatively and quantitatively different from those inhabiting remote from this influence in the soil environment. Therefore, the rhizosphere is a unique subterranean habitat for microorganisms. The rhizosphere microflora of one plant differ from the rhizosphere microflora of other plant. Thus, rhizosphere microorganisms differ plant to plant both qualitatively and quantitatively. Rhizosphere region can be divided into two zones. The inner rhizosphere, which is in a close vicinity of root surface, and the outer rhizosphere embracing the immediate adjacent soil. In 1949, Clark has suggested to use the term rhizoplant for root surface itself in studying the rhizosphere phenomena. Now next is why microbial activity are more in rhizosphere. Generally, the number of microorganism or microbial population in the rhizosphere is more because the root release some exudates like organic acid, vitamins. So these exudates attract the microorganisms. Therefore, the microbial activity are always more in the rhizospheric region. Next is what is rhizosphere effect. The rhizosphere is a zone of increased microbial community as well as microbial activity influenced by root itself. However, this influence can be measured simply by plating technique and expressed a rhizosphere effect, that is a stimulation that can be measured on quantitative basis by the use of rhizosphere is to soil ratio obtained by dividing the number of microorganisms in the rhizosphere soil by the number of microorganisms in the non-rhizosphere soil. The RS ratio, that is rhizosphere and non-rhizosphere ratio, sensing with different growth stages, it is apparent that different types of microbe dominate at the particular growth stages. A single microbial species will have to compete all the time to become a permanent inhabitant of the rhizosphere region, which is rather impractical. This microbial selection during different growth stages and sensing scenario of microbial community in rhizosphere or rhizoplant are ultimately governed by the root exudate of the host as well as the result of microbial interaction.